Today, I'd like to share our preliminary work on this efficient vaccine ontology mapping via the TF-IDF Retrieval and lightweight fine-tuned reranking. Yeah, this is our full co-authors. Yeah, this work was conducted between Dr. Ho's lab and Dr. Tao's lab. Yeah, and Scarehead. So the main points. Yeah, the goal was to we like to develop an efficient and reproducible vaccine ontology mapping pipeline, combining the efficient TF-IDF retrieval with the lightweight fine-tuned reranking using open LoRA models. When we conducted this research, because based on our previous research, like we tried to different approaches to focus on this reranking problem, like a cascaded framework or like just a large language models ranking, something like that. But it seems the available approach either depends on like computational resources or depends on a proprietary a system or like a GPT-4 this cost or something like that. So we like just to provide an alternative, is efficient solution to show whether we can utilize those small open fine-tuned learning model to do this mapping and re-ranking work, but it can also get this similar or better performance. Yeah, this is the main idea of our work. Of course, yeah, this is just a preliminary findings. Our ongoing experiments with more learning models aim to extend this efficient ranking framework. Yeah. And for the modification and background, as we know, yeah, vaccine names normalization is the critical task. It will ensure consistency across the clinical trial registries and facilitates the integration with ontology-driven analysis. And the community ontology, like the vaccine ontology, provide a framework for interoperability and semantic consistency. Vaccine names, however, is varies widely, like trade names, abbreviations, manufacturer label, etc. And they prefer the work like cascaded fine-tuned mapping and element-based ranking improve accuracy, but we're computationally heavy and API dependent. So this is the challenge and the gap for this work. Vaccine name variability and insufficient contact information cause the mismatches in ontology mapping. For example, when we extract the vaccine names from clinical trials, it may just one word or two words or several terms without contextual information. But our ontology, like vaccine ontology, it may contain 6,000, more than 6,000 vaccine candidates. So to exactly match this vaccine names or interventions from clinical trials to the 6,000 vaccine names in the vaccine ontology is challenging. Yeah. And the traditional TF-IDF retrieval is scalable but less semantic depth. The large proprietary learning model like GPT-4 or Gemini improve context reasoning but are expensive and close to source. There is a need for an efficient, reproducible, and cost-free pipeline that balances performance and transparency. Yeah, the objective, develop an efficient and reproducible vaccine ontology mapping pipeline. And we conducted in two stages. In the stage one, we utilize the TF-IDF retrieval to narrow the 6,000 VO vaccine concepts to a top 200, for example, 200 or 300 or 100 candidates per mention. Yeah, this depends the candidates' performance. Sometimes that, yeah, we just utilize the 200 top candidates at present. And the stage two, we will utilize the learning model or the fine-tuned open learning model to reorder or re-ranking the candidate to find the best match, especially for the accuracy at the top 1. So yeah, we try to provide a cost-free alternative to proprietary learning model while achieving comparable or higher accuracy at top 1. This is the overview of our pipeline. It's a two-phase efficient vaccine ontology mapping pipeline. From the clinical vaccine names mentions, go through the two-phase vaccine ontology mapping pipeline. First, phase 1 is TF-IDF plus role-based retrieval. Then level the 6,000 to top 200 vaccine candidates. Then in phase 2, currently we apply two strategies. First is the GPT-4 reranking at the baseline, and the second is our fine-tuned Jianfu 3 reranker. Yeah, fine-tuned with the LoRA. Then finally, we have the final mapping and do some evaluation. So figure 2, this figure shows the DFT, the parameter efficient fine-tuning of this Jianfu model with the LoRA. So we can see here the training input, we input just the vaccine mentions from the clinical trials and the top 200 TF-IDF candidates. Yeah, then it will be contained well on definition, trial metadata, etc. Then move forward to this transformer Jianfu model. It's only 0.6 billion parameters. It's much smaller than the available learning model like GPT-4, etc., or Lamar or Gemini. For this LoRA fine-tuning, we can see that the base weights are frozen, like those WQ, WK, W, those matrices are frozen, but we're only using the LoRA to insert or inject the adapters like for two layers, the self-rotation layer and the feed-forward network layer. And the training only of this, the matrix, the up projection and down projection, matrix A and B. So finally, we get the results. Then the output will be the candidate survey scores and the ranked vaccine ontology concept, the top key. Currently we keep the top 200 used for re-ranking the TF-IDF candidates. That means we based on the 200, then we have the top 10 or to calculate the performance of top 1, top 2, top 3, etc. to see whether this fine-tuning with the LoRA can improve the final top key performance, especially for the top 1. This stage 1 TF-IDF Retrieval is use lexical token and synonym-based matching against the vaccine ontology. Retrieves top 200 candidates for curious for recall coverage. We like to get a higher recall coverage. So we choose the 200 candidates as our first stage results. Then it will provide the fast, interpretable, and scalable retrieval layer. For example, this term from the 6,000 level from 6,000 to this one, the currently included within the top 200 candidate list. Then we use the fine-tuned or low-level model re-ranking, using contextual ranking to refine the TF-IDF candidates. We compared it to model 1 the GPT-4 zero shot. Since GPT-4 provides strong reasoning, but it's costly and closed model. We cannot to get their width or something like that. For the Jianfu 3 re-ranker, we chose this model because the first is initially pre-trained on those re-ranker or fine-tuned on the re-ranker. It mean different domain. And it's a small model and it's open width. So we just fine-tuned based on this model. And in fact, we also tried using the Jianfu 3 re-ranker 0.6 billion model, zero short, but the performance is very low. It shows the domain matters. So we fine-tuned using our domain knowledge, then improve the performance. Yeah, this is our data set. We have 550 vaccine mentioned from clinical trials, mapped well annotations. Each entry contains, like mentioned, candidates, trial metadata, and authority definition. We evaluate using the accuracy and MR mean reciprocal ranking. Yeah, we compare the baseline TF-IDF, TF-O, Jianfu model, and the TF-IDF limited to top 200. So this is the result. We can see here the fine-tuned Jianfu achieved the best top one accuracy. And the TF-O, zero short is only 0.56. This TF-IDF is the baseline, is our initial stage 1 matching. Then TF-O fine-tune or based on the 200 candidates from the further stage. Yeah, to the phrase. But we can also see that TF-O is a top 10 performance is better than Jianfu. And the MR matrix can also see fine-tuned Jianfu achieved also the best MR results. Yeah, this is just our preliminary findings. So this is the key findings and the discussion. The fine-tuned Jianfu surpasses TF-O by 13 points in top 1 accuracy and 10% higher the mean reciprocal ranking matrix. And the TF-IDF plus the LoRA tuned open element can achieve the scalable or can be a scalable reproducible solution. It also demonstrates that efficiency and openness can coexist with high performance or similar or just comparable performance. So this is the conclusion of our future work. The TF-IDF plus fine-tuned Jianfu ranking pipeline improves accuracy, transparency, and cost efficiency. It shows the open source model can rival or outperform proprietary APIs, fully reproducible, lightweight, and extendable framework. Ongoing experiments with more LLM models. Yeah, we will integrate contextual information to enhance prompt tuning for elements in the future. Yeah, so thanks for the project members from Dr. Ho's group and Dr. Tao's group. Also thanks for the project funding from NIH. Thank you.
Hi, great, Jeff, for the talk.
Yes, thanks.
So, any questions? So, like Jeff, like you show like you have different methods, right?
Yeah,
I see that's like one result selection. You show like the this one. You show the Quen 3 fine-tuned one is better than TF-IDF.
TF-IDF is our initial mapping. We just utilize the since it's TF-IDF, it can be fast and flexible, and can combine with a reward-based something like that. So we just try to utilize the TF-IDF to provide an initial mapping to narrow the 6,000 fast names to 200 top 200 candidates. So we also did the experiment like try to just utilize the LoRA model to directly mapping, but find it's harder to implement since the 6,000 fast names, the token input will be much larger than the limit of the LoRA model. So that is why we try to utilize these two phases. First, we use an efficient TF-IDF to do a initial mapping to narrow the 6,000 vaccine cohort to or ontology to a 200 candidates. Then it will provide a efficient and the more inputs for the LoRA model. So for 200 candidates, we can easily fit to the LoRA model. To GPT-4, to Jianfu, then we did the stage 2 experiments. Firstly, we utilize the GPT-4 as a zero shot. Then we utilize the Qiang 3 for fine tuning with LoRA. Yeah.
Okay, great. Thank you for explanation.